Today we discuss so-called principal component analysis, PCA. The purpose of this procedure is to encode the information contained in large datasets, large in a sense that we have many variables, into some smaller number of variables that can later be used for something. For example, the new variables can be used in ANOVA, or new variables can be used in multiple regression model, or usually principal component analysis is the initial state of the so-called factor analysis, which will be the subject of the next lecture. Principal component analysis is an exploratory technique. We usually do not have any hypothesis at the very beginning. We just look at our variables and try to make sense of them, try to reduce the information to the simpler form. From the mathematical point of view, the new variables are linear combinations of the initial variables, which means that our initial variables should be of numerical kind, in such a way that linear combination makes sense. Principal components, again, from mathematical point of view, are just eigenvectors of correlation matrix or eigenvectors of covariance matrix. So let us first look at some example how this technique can be used. Our first example is the following. Paleontologists analyze results of measurement of some number of teeth of certain extinct mammalian insectivore. These teeth were found while quarrying limestone in some place in South Wales. And paleontologists supposed that there are at least two, maybe more, species of similar animal, and to distinguish between those, to check whether it's true, and to distinguish between those two species, they collected the teeth and they made several measurements. And here you have a picture of just the scheme of the tooth and some measurements that were made. So as you see, the shape is quite complicated. Therefore, you can assign several numbers to each tooth. And here there is some artistic view of the possible these animals. Okay, here you can see 1, 2, 3, 4, 5, 6, 7 of the measurements. In principle, there were 9 of them. So then the correlation matrix between all those measurements was composed. The matrix was diagonalized, and 2 of eigenvectors were selected as principal component 1 and principal component 2. And here we have a scatter plot of all these teeth from 1 to 88 on the plane of PC1 on horizontal axis and PC2 on vertical axis. And what was the conclusion? The conclusion was that since they seem to cluster there is the pink cluster here and there is the blue cluster here. At least there is a difference between those two groups with respect to PC2, so the second principal component, so the second eigenvector, which is the first and which is the second, and why they are ordered and numbered, we will see it in the moment. Therefore, the researchers concluded that there are two, indeed, there are two species, and the pink cluster represent teeth belonging to one of the species, and the blue cluster correspond to teeth belonging to the other species. The interpretation of those components was based on correlation of values of the correlation of the components with the original variables. And for example, the first component appears to have positive correlations, positive and comparable correlations with all the variables. Therefore, this first component on the horizontal axis today here was interpreted as containing information about the average size of the tooth. And the second component had negative correlations with all the length variables. So here we have one length variable, the other, and yet another length variable. And it had negative correlation with the rest, so with those vertical on this picture, those vertical variables. Therefore, it was interpreted as containing information of the size of not the size but the shape of the teeth. So it could, in some sense, distinguish between teeth that are rather elongated and teeth that are rather round. Yeah, and this shape on the vertical axis here appears to distinguish between the two species. So let us now, we don't have the data for this experiment, let us now perform such an analysis for the data that we do have. And this will be a very standard data set containing information about shape and some measurements about shape of the flower of a plant called iris. In Polish it is also called iris or kosaciec. So to know what we are talking about, let us refresh our memory about the structure of the flower. So here we have some here is this iris flower, and here is some other flower. If we look at the iris, we could probably, not being botanists, we would all the purple parts of the flower we would classify as petals. However, it seems that only some of them are petals. These are petals and these are sepals. So to distinguish between petals and sepals, we can also look at some other flower. So these colorful parts are usually petals, and these green parts here are sepals. However, iris have all of them are colorful. Okay, so what are our variables? The variables are the following: sepal length and sepal width. So we measure how long it is and how wide it is, and the same about petals, how long it is and how wide it is. And we have the fifth variable. In this data set, we know what are the species. So these are these three different kinds of irises. In our data set, there are 150 flowers. What we are going to do here, we are going to perform principal component analysis and try to check whether we are able to separate between different species, looking at values of principal components. So what we do, first of all, we of course import the data, and another important thing is that we transform our variables in such a way that they all have mean value 0. It is important from the theoretical point of view. When we were discussing multiple regression, we were interpreting correlation matrix and covariance matrix as a matrix of some scalar product defined on the set of variables. And here we do the same. We interpret our covariance matrix as some bilinear, as a matrix of some bilinear form defined on the set of variables. And to avoid to have this matrix to have this matrix defined on the space of variables in such a way that this is not degenerate, we have to say project all the variables on the not project, maybe move all the variables in such a way that we just move from the affine space of variables into vector space of variables, choosing the origin. And the origin is in the place where all the means are located. And then we really have a bilinear form defined on our set of variables. So here is the correlation matrix, the covariance matrix of all the measurements, all 4 measurements. And what we are doing from the mathematical point of view, we are calculating eigenvalues and eigenvectors. So here we have eigenvalues. And now about ordering. So in this analysis, we order our eigenvalues and our eigenvectors from the highest eigenvalue to the lowest eigenvalue. Since our covariance matrix is symmetric, we know by spectral theorem that it can be diagonalized. Moreover, since diagonal elements are variances, and this is true in whatever basis we write down the matrix in principle. If we do it in after diagonalization, so if we take the basis that diagonalizes the matrix, on the diagonal we will have variances of the variables. Therefore, it is positively definite matrix. It always has positive eigenvalues, and we order these eigenvalues from the highest to the lowest. The sum of all eigenvalues is equal to the trace of the matrix, and this is interpreted as the total variance contained in all the data. What we want to do, we want to choose smaller number of variables, those principal components, so eigenvectors, in such a way that we don't lose too much of the variance. Therefore, we start from the top. And when we stop, it depends on our decision. Here we have, before we discuss choosing principal components, let us look at eigenvectors. This is the first eigenvector, the second, the third, and the fourth. And these are coefficients in the original basis. So if we want to calculate the value of the principal component, for example, principal component 1, we have to multiply first variable by this number, second variable by this number, third variable by this number, and the fourth variable by this number, and we get the principal component 1. And the same for other principal components. Okay, so now we choose our principal components for the rest of our analysis, and this can be done in the following way. Let us look at the plot of values of eigenvalues, but calculated as the percentage of the trace. So for each eigenvalue, I check what percentage of the whole of the total variance it contains. So we can see here that the first component has contains almost all variance, and the rest are really very very small. We can maybe add here a cell in which we will create the table with those percentages. So instead of eigenvalues themselves, I will put here the percentages for the picture. So we will see not only the picture but also the values. It is dangerous to do it during recording the lecture, but let us see. I hope it would work. Yeah, it works. Okay, so as you can see, almost all the variance is contained in the principal component 1, and only 5% in the principal component 2, and almost 2% if in principal component 3, and the principal component 4 gets very small percentage of the total variance. Therefore, for the rest of the analysis, we will take probably the first two principal components. To really choose how many of principal components we should take, we look at the plot. So here is the first, the second, the third, the fourth, and usually when we see a kind of angle, this is the place when we stop. So the angle appears at principal component number 2, so we should take principal component number 2 and 1 and principal component number 2. Or we can look at the cumulative percentage plot, and maybe this picture is not the best because the x-axis, the vertical axis starts from 0.9 or something like that. It is better to look at this picture because now we can see that really the first two principal components contain almost all the variability that is in this dataset. We will sometimes also look at the three principal components. We definitely disregard the four of them. So then we calculate those components. So we create the variables, and we will look at some pictures. So first, there are histograms of the original variables. So these two are about sepals. These two are about petals. As you can see here, we have a kind of bimodal histogram. Probably there is a difference between the species, maybe, and here we have 50, and here we have almost. There are some rogue ones. So probably this is just one of the species, yeah, this 50 flowers. About the sepals, the distribution is more similar to normal distribution. So there is no that clear difference between the between the specimens, species. Okay, so then let us look at scatter plots. They are color coded. We can see here the difference. We can see here what are the species. So those three species are color coded, and we can look at the scatter plot of sepal. There are misprints everywhere. Of the sepal length and width, and we see that those two variables separate this cluster, but definitely do not separate the other two species. The pair sepal length and petal length is better because we have a more or less clear separation between those two species. Always the blue ones are different. Definitely there is one species which is very different, and the two which are not that different but also separated. And we can just look at different scatter plots. Maybe we could just choose two of the original variables. I want us to look at those pictures to compare it to the appropriate picture for our principal components. Okay, so this is the end of the pictures, and now we can look at histogram of our principal components. And again, we see that the first principal component clearly separates between one of the species and the rest. And two other, I also took into account principal component number 2 and principal component number 3. They have more or less normal or similar to normal distribution. So they are not that probably they are not that good at separating species, but this may be good. So we have this scatter plot of the two components. On the vertical line we have the second component, and on the horizontal line we have the first principal component. And again we have this blue cluster. This is definitely separated from these, and it is not that easy to draw the line somewhere here, but still we can see that these two species can be separated somehow. We can draw the line somewhere here, or we can draw the line like that and separate those two species, and only small percentage of them will be classified wrongly if we use some separating line somewhere here. We can also look at the scatter plot of the three components. It is probably a little bit more difficult to visualize because we have here three different variables, so it is a three-dimensional picture, but again we see that we have some clusters here, so probably if we use the information about three components, we will be able to separate all those the three species. What are the interpretation of our principal components? To try to interpret them somehow, we calculate correlations of each component with original variables. And here we have the table. So our two components, our original variables. When we see at the column related to the first principal component, we see that it is negatively and highly correlated with sepal length, and petal width. And the correlations are really very high. So in a sense, it is correlated with more or less the size of the flower. Yeah, only with the correlation with sepal width is much smaller, and the second principal component is about sepals and not petals, because it is not the correlation between petal length and petal width is very small. Correlation between sepal length and sepal width is bigger, and the biggest with the sepal width. So we could either think of the first component as saying something about petals and the second saying something about petals, or think of this first component as saying something of the overall size of the flower, and this second principal component as saying something about sepals. Okay, so in our, as you can see, there is this interpretation is kind of not that clear thing, and it depends in some sense on the decision, what we how we see the data that we obtained. We should probably think of yet another problem here, which is not very important in our example. In our example, we had four variables that were measured on all in centimeters, and all the sizes were comparable. When we look, let us look at the histograms. Here we have numbers from 0 to 8, from 0 to 4 to 5, yeah, from 0 to 7, from 0 to 3. So very similar numbers. So the scale of all the variables is more or less the same. However, it may happen that we have data set with very different variables measured in really different units and very different scales. And then if we want to get rid of the problem of units and problem of scales, we should standardize our variables. So instead of the original variables, we use variables after standardization. So we not only subtract the mean, but also divide by standard deviation. And then proceed in the same way. calculate the covariance matrix, which accidentally is the same for standardized variables. It is the same as correlation matrix for the original variables, and what we get is and again we do the same. We diagonalize the matrix, then everything is measured more or less on the same scale. So let us see what we get in our example. So the picture is quite similar. Again, we have two important principal components. Principal component 1 definitely separates this variable, and here we have a little bit murky picture, but again, maybe now this line that separates the two species will be more vertical, and again there will be few flowers wrongly classified. And when we see the three-dimensional picture, this is more or less the same view. When we look at correlation with original standardized variables, so here I've written sepal length, sepal width, petal length, petal width, but I have in mind standardized variables. So the correlations are very similar. There are differences on the second or third place after comma, so nothing much happens here. We can compare the pictures. Maybe I will make them a little bit bigger. Yeah. So you can see that really the separation line here is rather vertical. Here I will use a line like that, and here I will rather use a vertical line. So it is much more visible on the standardized version that it is really the first principal component that separates those species without using the second principal component. Okay, so this is all for this lecture. In the next short video, I will show you the example of principal component analysis performed on the data set that we used before for multiple regressions, for selecting variable for multiple regression. I mean predicting life expectancy. We will see whether we can, using principal component analysis, whether we can create variables that can be used as independent variables in multiple regression to replace the original variables that we selected via our 4 different procedures of selecting variables for multiple regression. But this video will appear provided the analysis will make sense. This is just the idea that I got to use this principal component analysis. But since it is an exploratory technique, it may just not yield important information. So that's all for now.